All right. I hope we can start the meeting. Could someone confirm if I am still audible?
Yes, Hasidi, you are audible.
Yes. Good evening, everyone. I am Harsiddhi, President of International Society of Computational Biology, Student Council, Regional Student Group, Tamil Nadu Node. It is my pleasure to welcome you all to our sixth webinar titled Standing on Solid Ground, Navigating the Evolving Landscape of Bioinformatics with Insights from Epigenomics. We are delighted to welcome our distinguished speaker of the day, Mrs. Midhuna Diez, Bioinformatician and PhD Candidate at Saarland University, Germany. Before we begin the event, I would like to briefly introduce our organization. The ISCB-SC RSG-India is your local window to global opportunities in computational biology. ISCB is a global organization based in the USA that drives computational biology forward through research, training, outreach, and community building. The ISCB Student Council, started in 2006, nurtures the next generation of computational biologists with RSGs in over 26 countries. RSG India connects and promotes a strong nationwide network of computational biologists linking local talent to global opportunities. We proudly collaborate with premier institutions including Mahindra University, NIT Calicut, Manipal Academy of Higher Education, IIT BHU Varanasi, and Srihar from Tamil Nadu. Here is the RSG India Core Committee for the year 2025 to 2026, supported by faculty advisors and visionary leaders across India. RSG India has successfully organized impactful events in the past, fostering collaboration, learning, and innovation in computational biology. Here are pictures from some of the events organized from 2023 to 2025. You can reach us through our website or connect with us on social media platforms. Now over to Abdul Aziz, Secretary of the Tamil Nadu Node, to brief about the student initiatives at Sri Ramachandra Institute of Higher Education and Research.
Thank you, Hasidi. Good evening, everyone, and a very warm welcome to everyone that have joined us this evening. I'm Abdul Aziz and the Secretary of Srihar Node of ISCB RSG India. Established in 2025, we are a student-driven community with a vision of creating a vibrant platform for aspiring computational biologists and bioinformaticians. Our goal essentially is to create a platform where bright minds can collaborate with researchers and industry professionals through events, workshops, and webinars, thus encouraging learning and collaborations. Next, I'd like to introduce the team behind this effort, consisting of members from both undergraduate and postgraduate levels across all of the years from the Department of Bioinformatics. This is a picture from our first successful event, introductory event held on campus at Sri Ramachandra Institute of Higher Education and Research. I'd like to give a special mention to the Ministry Board, Dr. John Davis and Dr. Abhinand P.A., Dr. Arundhati M. from the Department of Bioinformatics at Srihar. Speaking of which, established in 2003, the Department of Bioinformatics at Sri Ramachandra Institute of Higher Education and Research offers both BSc and MSc in bioinformatics with a comprehensive curriculum blending the basics of biology and technology. We focus on hands-on learning, interdisciplinary approaches, while being on par with the latest developments in computational approaches in life sciences. Students from our department have gone on to make a difference in leading companies across the globe, such as Pfizer, AstraZeneca, EMBL, and Clarivate, and also into leading academic institutions such as University of Cambridge, TUM, the University of Saarland, University of Dundee, etc. And we are very proud to mention that we've previously had many of our alumni on our platform as guest speakers to share their valuable insights with us. And then I'd like to give it back to Harsiddhi to introduce the speaker of the evening, Mrs. Midhuna Diez.
Today, we are incredibly honored to introduce our guest speaker of the day, Mrs. Midhuna Diez, as mentioned, Bioinformatician and PhD Candidate at Saarland University, Germany. She specializes in the epigenomics of rare and infectious diseases at the Mueller Lab Department of Genetics. Her research leverages computational epigenomics to uncover the causes of rare disorders with unknown genetic origins. She also works on mapping the long-term impacts of malarial pathogen exposure on the host immune system in collaboration with labs at Stanford University. Mrs. Midhuna holds a BSc Honours in Biomedical Sciences from Ramachandra Institute of Higher Education and Research, Chennai, and a Master's in Bioinformatics from Saarland University. She draws on this dual background to develop and apply methods for interpreting complex genomic data and uniquely bridging the gap between foundational biomedical knowledge and advanced computational application. Please join me in giving a very warm welcome to Mrs. Midhuna Diez to begin today's session. A quick reminder, please stay until the end to fill out the feedback form which is required for your participation certificates. Thank you all. And over to Mrs. Midhuna Diez.
Thank you, Harsidi. Could you hear me all right?
Yes, ma'am.
Okay, great. I will share my screen first.
Yes, ma'am.
Do you see my screen all right?
Yes, ma'am.
Okay, great. So good afternoon and good evening to all attending. I am very thrilled to give today's talk. And first and foremost, I think I should start by thanking Dicky sir and the committee for extending me the invitation to talk today, and also to Harsidi for coordinating it with me. And I wish you the best to you and your team. You've done a great job. So yeah, I hope that the upcoming talks would be as productive as well. So I wanted to get started by explaining how I chose my particular title. We live in a world that is very fast-paced. To put it in perspective, what was in a matter of 5 years, we have something that wasn't there, which is AI. We have large language models that are constantly taking up our every second conversation at the moment. So the conversation go AI and something else. This is where we are at the moment. And the question again that usually people who might be starting, who are in their early careers, might want to know is which of these pathways that we have is the best suited one for me, and how to improve as a bioinformatician. So I must clarify and also understand from Harsidi is that many of you come from different backgrounds. So I understand that there are many who are in their early stages of their career, in their bachelor's or master's, and those who are considering to change career paths from wet lab into bioinformatics, or even some seasoned professionals, some professors and lecturers. So it is quite hard to cater a 40-minute talk across the spectrum. So I have taken a more slightly balanced approach. So I hope that you would be able to understand irrespective of where you fall in that spectrum, and also have something meaningful to take home. And if you have any questions, I would be asking in between. So please feel free to ask in between as well. And let's make this a little bit more interactive. All right. So first, I wanted to get started by emphasizing on this part that the fast-pacedness of bioinformatics has always been the case when it comes to bioinformatics. Bioinformatics is not a singular domain of science. It has been an interdisciplinary science, right? So if there is one of the interdisciplinary domains that has some sort of a development. Say, for example, there's a development in technology, a development in mathematics that has come. It cumulatively affects bioinformatics, and therefore we see a rapid progression in bioinformatics. And I would like to take this particular framework of sequencing technology that has developed over the years as my framework on which I say that in the past we have developed so much, and we are still developing even more, and that at the pace at which bioinformatics is evolving is very fast. So please keep in mind of the timelines that are mentioned here. So here we have, I will not be talking about every single year that is mentioned here, but I would go through certain ones. And starting with 1977, we have the Maxim Gilbert method that was published. And then over from 1990 to 2003, that's a whole 13 years, was when Human Genome Project was carried out. This humongous project, this milestone project that was done. And the way they did it was using a particular method called hierarchical shotgun sequencing method, where the entire genome is broken into manageable fragments, and then they were sequenced into small fragments, and they were meticulously examined and put together, and then the whole genome was created. So think about the time and the energy, the consumption of it all was for creating the very first Human Genome Project. And now moving forward a couple of years, now we have 2005 and 2006, when Solexa or Illumina, which bought Solexa, developed its sequencing by synthesis method, where they produced a high throughput method where the genomes or different fragments, it doesn't matter how big the genome is or whatever it is, that they could be sequenced in a very short time. This high throughput, the next generation of sequences had come forward. And which means we were able to reduce this 13-year-long requirement for one genome to a matter of weeks and days, and it was developed even further. So with each milestone that we reach, we also have years in between and different people coming together and trying to improve the accuracy, the efficiency of a certain technology. So to put it into perspective, maybe we can use the analogy of smartphones. So before we had this Nokia 1010, whatever it is, with the snake game that we all are familiar with. That was considered as a phone. And then in 2007, Apple dropped its first iPhone, and then we saw that it came with a touch interface, with a camera, with all sorts of features. And then phones were never the same afterwards. And what happened afterwards were different companies, different people came together to improve the technology, improve the accuracy, the efficiency. And with all improvement, with all the features that were put together, it also resulted in reduction in the cost. So it became more affordable in some sense. And so one can get all the features that was available in iPhone, but now can also be taken by another company for a very cheaper price. And in the same fashion, at one point, there was another invention. They dropped biometrics, for example, and then the phones were never the same. They all started including biometrics into it. So the same principle can be applied in terms of understood for sequencing technology. With each improvement or each milestone that we reached with sequencing technology, the cost of producing the sequences, right, the genomes, also dropped, because the sequencing machines became more affordable, the multiple labs around were able to buy, and therefore, yeah, so therefore there was an explosion in terms of the number of sequences, number of genomes, and samples that were sequenced over the years. We moved from when GenBank was created in 1990s, we see that it moved all the way in 2024 up to 1 billion entries. That's a huge increase. And we should remember that this information is available. This is an explosion of information that is available primarily because the time that it takes to sequence has been reduced significantly. Now, it's not just the time that has been reduced, it is also the size of the machines. Previously, there were huge rooms that were required to host these machines. Now, we have in 2024, Oxford Nanopore Technology dropped their Minion, which is a sequencing machine that can fit in the size of the palm of your hand. It can fit in your hand and can be transported anywhere. And not just the size of the machine has reduced considerably, they also improved on the size of the fragment of the genome that were, or RNA or DNA, whatever that is being sequenced. It moved from 100 to 300 base pairs to 10,000 to 100,000 base pairs. That's a significant improvement. So we went from short reads to long read era, where we can, instead of doing a jigsaw puzzle of 1,000 pieces, we can deal with 9 pieces instead. So if anyone who has worked, if there's anyone who has worked on jigsaw puzzle, you must know how time consuming it is, first of all, to put together all the information. And secondly, there's always information gap. You might fix certain things and then you realize that you still haven't figured out the rest of the puzzle over here, and then you move on to another corner. And there's always a misplacement of a particular jigsaw puzzle. You might lose one piece over here, or you misplaced it somewhere else, and it gets very complicated to figure it out. Whereas when we deal with in a bigger size pieces, it's easier to fit. And it's the same principle for ONT data as well. Long reads reduce, or they address the problems of information loss, reduce the problems of alignment issues, and so on and so forth. So these are some improvements that were done over the years. And then it was not just there. And in 2018, ONT dropped Promethean, which is also, it addresses even further of all the next generation sequencing related. And it's in fact, now we've moved back from next generation to nanopore sequencing methods. And yeah, it's a countertop method where it's high throughput and long read that can be done in a matter of days or even hours. So we have moved considerably from taking 13 years for a genome to a couple of hours, and from small fragments of data that is 50 to 500 base pair long reads to 100,000 base pair, 1 megabyte megabases long reads. So that's a huge improvement in terms of bioinformatics. And with that, we also have new technologies, new tools, new data sets to be taken into account. Now, it's not just the length and the time taken that has changed. It is also the resolution of your data that has changed. And we can question how one wants to approach their study in 3 different directions or 3 different ways. Say, for example, I want to study this brain. I want to understand what's going on, what's going on in the brain between a healthy donor and a person with certain disease. And I want to understand that. So in order to get this analogy even better, I'm going to change them into Lego brains. Okay, so it's easier to understand it. So we have this Lego brain, and we want to understand what's going on between the healthy patient, healthy sample and the patient with certain disease. So what one usually does is to take the tissue, dissociate the cells, take the RNA. Let's talk about transcriptomics. They will isolate all the RNAs. They will make the library, sequence it, and then try to understand it. So they will try to understand. Okay, here we have the expression profile of healthy samples, and here we have the expression profile of the person with some certain disease, and let's do some statistical analysis, try to compare these two and see what's going on. We can do that, but we also know that certain expression changes are not ubiquitous. It's not happening across. It's not necessary that the whole of brain has a change in the expression. It's maybe certain local areas, certain pockets of areas that needs to be studied. That's where we have transcriptomic level studies. We're like, okay, so this is the area where there's certain changes that's happening. Oh, this is the area where the other change is happening. We can try to understand it from that perspective. Or we could also try to understand it. What are the cells that are actually driving those changes? Is it all the cells in that region, or are we talking about just oligodendrocytes or glial cells or whatever it is? So we have another layer, which is the single cell aspect. So each of these data give us different information. So if you have the bulk data, you can have an overall profile, but the problem is that it doesn't have the cellular resolution that we're looking for or the spatial resolution. It just gives you overall profiles. Whereas when it comes to spatial data, you can understand it. Oh, these are the places where the changes are happening, but you just don't know what cells are involved. Whereas in the single cell data, you know exactly which of the cells are changing in certain places, but just don't know where they fall in the tissue. So each of these data sets have their own pros and cons, and they also come with their own sequencing methods. And there's also a cross sequencing method as well. So it is important that one is aware that these kind of different ways of studying are available. And importantly, this also points to the requirement of integration of different data, which I will talk about in some time. Now that we have talked about the bulk to spatial sequencing, it's not just the sequencing method that has been developed. We also have different biological layers. So we have the multi-omic perspective that we could study. We could study the genome, the DNA, the changes, the SNPs, and all the indels, all the changes that's happening. Or we could study the transcriptome, the RNA, and the small different types of non-coding RNAs, long-coding RNAs, and all sorts of long-coding RNAs, and so on and so forth, and how they change, the profile change. And then we can also study the proteome, which is the fundamental unit of the functional unit, and we can study how the proteins are changed or how they are produced and all these things. But then we can also study something that is above them, which is the epigenome, which is the regulation of the genome and transcriptome layers and how proteins interact with genome. So all these things are interconnected, and each of them can be studied individually as well. Now, I want to briefly introduce epigenome for those who might not have heard of it, so that we are all on the same page. So a gentle introduction would be that we all know that the body, the information is contained as DNA, and the DNA is also packaged in the cells as chromosomes. This is a very basic overview I'm giving. And this DNA is not just the way it is packaged into chromosomes is with the help of histones. So the histones are proteins that wrap around on which the DNA is wrapped around, and they come together. They sort of package together into certain structures, scaffolds, and many others. And then all these structures pack even densely, and then come together to form chromosome. And depending upon the accessibility, for example, that is how close these histones are or how far away these histones are, the accessibility of these DNA is affected. So here the histones are a bit spread apart. Therefore, this stretch of DNA is accessible towards transcription. And the DNA itself could have certain chemical modification, like methylation, that has an effect on it being transcribed. And then the third thing is it could be that the histone, the protein itself, has some sort of a modification on its histone tail, acetylation, methylation, and so forth. And those can also have an effect on whether or not a particular region is accessible or is transcribable. And based on that, we can design certain studies, chromatin accessibility studies, to see if it is accessible or not. Then histone modification study, what kind of histone modification affects, because these histone modification has an effect on the promoters and the enhancers, different parts of the genome. And then we also have the DNA methylation. How does the DNA itself allow for certain transcription factors to bind to it and cause the transcription process? All these things can be studied. Now, remember that I'm still talking about the epigenome layer alone. So you can see the intricacy of it all. There are so many other layers that are involved. And epigenome alone can be taken and studied from bulk or single cell level or a spatial level, all these different things. And usually, what one does is not just take this epigenome alone and then study it, because, like I mentioned, each of these layers are crosstalking. So we need to understand this crosstalk in order to understand the overall biology. So that's where integration of data comes into play. That is very important, because integrating different data actually improves the confidence that one might have in terms of what they are observing. Because if one sees a change in accessibility, but there's nothing happening on RNA or something, it could be that it's just random, or it's there is no way one can be sure about it. But integrating different layers, different components of the same layer together makes it more convincing, and you can actually see a pattern. You can see how it all works together. For example, here I've pulled it out of a pulled this particular image from a single cell paper, and you can see that there are 3 samples from 3 different people, and then each of the samples have a certain number of cells that have been sequenced. And here they have what has been sequenced is the transcriptomic level. So it's the RNA information that we have here. So single cell RNA-seq. And what one could do is to compare between these 3 different people. You have sample 1, it has some sort of a disease. Sample 2 and 3 are controls, and we're trying to understand what's going on. And this kind of research, or this kind of a questioning, or this kind of an integration is called the horizontal integration. And then you also have the other kind of integration where, like I mentioned, you go across different omic layers or different parts of the same omic layer. We take their methylation, we take the accessibility, and we take the RNA, and we try to understand what's going on in all of it. So on the methylation level, at the promoters, we see that there is less methylation, and then we also see at the accessibility level, we see that there is more accessibility, and then we see that there is an increase in the number of RNA, the expression. We can sort of see the story come together, or the other way around as well, where there is more methylation on the promoter region and the accessibility is closed, for example, and then there is less methylation that is less expression of the data. Then we can say that, okay, this particular gene is not being expressed. We don't have any information over there. So you can try to understand it from all these different blocks. Or one can also do an integration by diagonally, where the samples come from different places, different individuals. At the same time, the information is also quite different. So we have the methylation over here. Maybe I will change my thing to laser pointer. Yeah. The information over here is methylation, whereas over here is the transcriptomic information and the accessibility information, but they all come from 3 different donors or samples, and we're trying to understand it across. That is also possible, but it comes with its own set of limitation. All these methods come with their own set of advantages and disadvantages, and that is where we have different pre-existing tools, different things that one might have to learn how to use them, which brings me to this humongous— Excuse me, ma'am. Yes.
Yes.
Actually, I had a question. Yeah. What is the difference between this vertical and diagonal integration? I didn't catch it.
Sorry. What's the difference between vertical and diagonal integration? So here, what we see here. Okay, let's start with horizontal. All right. Um, Horizontal. I am assuming you understood. Is that right?
Yes, ma'am.
Okay. So let's start with, then we can go on to vertical integration. Vertical integration is that it's the same set of cells. We have the same set of cells that come from the same donor, and we have taken information for methylation, information for RNA, information for accessibility. All three of them come from the same population of cells, right?
Oh,
Whereas over
okay.
here, it comes from three different populations. And therefore it is not the same, but they come from three different, which means it brings about its own technical issues. Like, for example, one might need to consider the technical issue of batch effects or something of that sort. So it comes with its own set of problems, but this is another kind of integration.
Okay, ma'am. Got it. Thank you.
All right. Yeah, pleasure. All right, so like I was mentioning, there are different types of integration so far, and after these integration, we have to consider what kind of software one would want to use in order to do these integration, and that's available in plenty. Sorry, it's not moving. Oh, there we go. Yeah, that's available in plenty. We see that there are so many different softwares. um, we have for the single cell part, for management of different libraries. We have it for RNA, single cell, bulk, whatever be it. There are so many different types. And I've also put in some programming languages that one should probably know, or should essentially know, and things like that. So there is so much of information that is available at the moment. So what we have looked at so far is that there are so many types of sequencing technologies, so many ways of looking at the data. There is a humongous, like a mountain of data that is already available. There is different levels that we can look at, from epigenetics to proteomics to all sorts of microbial biodata, whatever be it, metagenomics. There's so many different types that one can look into, and there's just plenty of options. But where does one start? And that brings me to my title today. What is standing on solid ground? What is this solid ground? What are the things that I would consider as essential, the foundation? And this landscape is been evolving over the years. There are always new tools, new methods, new machine learning models, AI, whatever be it. There's always something new that's coming up. Which ones are the foundation, and which ones are something that can be learned over the years afterwards, after the learning the foundation? And then I want to give an example from my own experiment, or the research consortium as well. So that's what the questions are. These are the questions under consideration. What are the new developments? We've looked at it so far. What is it that has been done so far? And it's much more than this. What I've given is just a small glimpse. And if you're already overwhelmed about this, great. But that means you've been tracking with me already so far. That's good. But the thing is, it's okay. That's the part. That's part of learning. That's part of this journey. And while it is can get overwhelming, it's also thrilling, because there's so much to look forward to. And then, so the next thing is what I wish I knew when I started my career in bioinformatics. This is a question I ask myself. Sometimes I think in retrospect, how things would have changed if I had known certain points, and if I had known them, how it would have changed my master's or my PhD, or how I would have chosen my bachelor's as well. So all these are I thought maybe I could share with you. And of course, after the presentation, we will have some time to go through some question and answers. All right. So the first thing is what I wish I had known when I started my bioinformatics path is that, so I think many of you might have come across this particular graph. It's a very famous one. It's called the Dunning-Kruger effect. And I wanted to share about my experience, which is when I started getting into the world of bioinformatics in my bachelor's. I did my final thesis with Dicky sir, and I was introduced to bioinformatics in my last year. And that was the year I was introduced to. I was learning things on my own. I changed my Windows laptop to dual boot with Linux configuration. And I was like, I thought I was on top of the world because it was so exciting, and I thought I knew what I needed to do. I was at the height. And then started my master's, and then I was like, okay, there's more than what I thought it was. There's much more than what I thought it was. And then I came into the peak of my master's. I realized I am not going to make this. It is hard, it is difficult, this makes no sense. And then slowly into my PhD, still doesn't make sense. What am I doing here? And then slowly they were like, oh, there are pockets of things that make sense. And then go back to, I'm never going to understand this. And then and then slowly I'm walking to the point of, okay, trust me, it's complicated. But then I go back again. So there's always this constant moving, swinging between, I will never going to understand this, to trust me, it's complicated. So when I say, I'm trying to explain, or at least I can put this one over here to show that as you grow in this field, you realize that there's much, much, much more than what you initially anticipated. And that's the case about any field in science, anything related to this world. So it's important that you are aware that there's much more, and there's always room to learn, and there's always room to collaborate. And this is something that I really want to emphasize, so that you are aware and not caught off guard, because that can happen so often when people apply for masters or PhD and realize, I am not supposed to be here. It's not about I'm not supposed to be here. It's about, what is your mindset? Do you want to continue on or not? And so yeah, that's the part I want to emphasize and say, have a good mindset. And I will go back to that again. So the next thing is the solid ground aspect. What is considered as the non-negotiables? And so what are the things that is absolutely essential? These things have to be irrespective of whether you're in a master's or in your PhD or in some sort of in an industry or whatever be it. You need to have certain skills as a bioinformatician. So we will go through that. And then the next best thing is always around the corner. There's a new AI tool, there's a new something, but are they just a fad, the phase, and then they would be gone? Or is it something that we really need? This is a question that you would, I encourage everyone to keep asking. It doesn't have to be that, oh, there's something new that has come up, the new, most innovative deconvolution method has been dropped and we should use it completely. No, not necessarily. It could be that you just use linear regression and that is sufficient, because you need to be able to explain your data. That's what matters. So I want to go into what it takes to be a bioinformatician. So I think these are certain key pillars, or the key points. I mixed two analogies there. Key points that I think one should keep in mind in order to work towards becoming a good bioinformatician. So what are the first thing I have mentioned here is the core foundation. What are the foundational things that I need? First thing is a solid background in mathematics and statistics. Bioinformatics requires math, period. It is absolutely essential. One needs to be able to understand all the papers one would be reading. So these are the points I wish I had known when I started as well, because I did not prioritize mathematics in or understanding the concepts of it when I was doing my bachelor's, for example. So I would say focus on it, because in order to read a paper, in order to understand the machine learning model that was used in order to get a particular data, you need to understand what that model does. And therefore, it's important that you invest time and energy into understanding the math, understand linear algebra, understand how different probability and statistics work. All these things are different statistical tests that will need to be done. All these things are important. Different machine learning models, the basic ones that are there, those are absolutely essential. What are those different things? What is isolation first? What is different types of tree, decision tree, and all the questions like what is logit function? What is like trying to understand these kind of concepts is absolutely essential. And then it's not just the mathematics, but biology. One needs to have really good, strong foundation in biology, because the question we are asking at the end of the day is a biological question. That's what differentiates a bioinformatician from an informatician or a mathematician. So you need to understand the concept of what is it that we're trying to do. So be data oriented, understand your data in and out, and see what's going on. You need to understand the concepts. You understand where it was collected, how it was collected, what are the drawbacks, what are the limitations and the potentials that we can see from the data that we have at the moment, and what kind of model that does it follow, what kind of population are we talking about. All these things are important, and whether it's in vitro, in vivo, whatever be it, all these things matter. And the third thing is informatics. Have a strong informatic computer science background. It's not just putting in 5 lines of code from ChatGPT. It is trying to understand what your code does and how optimized it is to process a huge amount of data. You should be able to understand how data set work, how servers are used, what is high performance computing and what it means to be working on high performance computing. All these things are important stuff. And it's a skill that you learn over the years with exposure. But if you build consistent habits of learning over the years, you would absolutely accumulate a wealth of knowledge. So I would highly recommend doing learning about these things. The second one would be the technical skills. What are the technical skills over here? Absolute essential thing is programming. R, Python, Bash, any other, there are new ones that have come up, Rust, and so on and so forth, Julia. And these are all so many different kinds of programming languages, but the most basic that is used by bioinformaticians is R and Python. These are the two major ones that are being used, and Bash is absolutely is important as well. So it is important that you have a good knowledge of it. Again, I would say it, ChatGPT can be helpful to make some use cases, some sort of a small 2-line boilerplate kind of a code, but they cannot be substituted for actual knowledge. One needs to know how to write one's own code, understand someone else's code, and have good coding practices. The code has to be reproducible. The whole workflow should be reproducible in order to get them published. So having a good system that is knowing about what is Git and pushing, and then repositories and things like that. These things are absolutely essential. Knowing about different pipelines, which of the pipelines are already available and which are the ones that are being made. For example, what is Snake, what is Nextflow pipeline, and all these things. Understanding these are important. And also when you write your own code, trying to know like what is the good coding practices. Each of the coding languages or the programming languages come with its own grammar, like how they should be structured, how they should be done, and things like that. And there are plenty of resources online. One needs to just keep going and understanding how it should be done, what the syntax is, how to write a proper code. I'm emphasizing on this so much is the primary reason is because we live in like I mentioned, an AI world, and I have seen it amongst people, generally students who have not had a good background in coding, and unfortunately that's the case. Like we all are learning, and there is nothing wrong about it. So as you learn, it's good not to rely on just ChatGPT brought in information, but rather understanding. It's not bad to use it, but understanding that writing your own code is absolutely essential, and like understanding that this is what is going to be published, and this information that you're producing from your code is going to be taken as gold standard in some sense, and it's going to be peer reviewed or not, depending upon what kind of thing you want to be sharing. And then that's going to have some implication. People are going to take it into understanding further based on whatever you create. So it's important that you write. So I hope that I have driven that particular point well. The third thing is thinking and mindset, which is one should be able to understand how to frame a problem, how to understand the data set. So again, I would say, know your data, know what is going on and how it is collected, how it has been sequenced, how it is been processed, pre-processed, or if you're pre-processing, you should know what kind of pre-processing step that needs to be done. And you need to know what kind of problems you might face, what kind of technical challenges. Like, it's always important to be curious about your data and to have some sort of a critical mindset, because just because a particular tool has been published doesn't mean that that tool is used across all sorts of data sets. One needs to understand what kind of tool are we using and why are we using that particular tool. It doesn't make sense for my data. Is there is there a problem with the tool itself? These are certain questions that one has to ask before using it, because it was already published somewhere. Or even if it is very famous, one needs to keep asking the question, what kind of the underlying assumptions are for that particular tool. And then the last thing is positive thinking. When it comes to research is extremely high risk. So one might go in full on saying, I will give my 100% and all my 1 year of research into this particular topic, and I will try to give my best, I will do this and that, and then realize that there's actually no significant results to give. And that's part of the process. That is research. Negative result is a result. And the question is, how do you take it, and is it a true negative result, or is it something that could be improved and you actually get some other result? So like, to be precise on that is important than to say that I should always get a positive result. So having a good mindset about that is important. And then the second last one is the software and management skills. As a bioinformatician, you are expected to communicate your data with biologists, to clinicians, to the world. And it is absolutely essential that one learns how to best represent the data. How do you plot your data? What kind of different plotting systems do you have? How do you generate different figures for your paper? All these things are important. So please take time to learn. These are all soft skills that are usually found. You can find workshops online or even offered by our own universities. Please take time to see how well to communicate different ideas, because at the end of the day, we're doing research so that we can communicate what we have found. So it is important to see how to communicate by figures, by in terms of presentations, and in terms of just using language that can be understood by other biologists and clinicians. It is important to form this collaboration. It is important to have a very collaborative system. Otherwise, it is not possible, because each of them have different we have expertise in different fields. One might understand certain concept better, but bringing all of them together makes it worthwhile. And then lastly, I would say it's important to have a project and time management skill, because the project, usually when it comes to high throughput data, they are really big and they can go in different directions. So listing down what your objectives are and then following through, you list down your major objectives, and of course there would be redefining that happens over the time, but listing them down and trying to stick as much as possible to that is absolutely essential. Otherwise, it is very easy to get sidetracked and it can go wrong very quickly. And the last thing I want to explain is the ethics and the data privacy. It is important that unfortunately, this has to be emphasized again, that ethics is something that is unfortunately not being promoted or like upheld in many places. Many researchers are found to have published wrong data, like forged data, or they have changed their hypothesis to suit the results that has come. So there's a lot of something that's called p-hacking, to make sure that statistics somehow it makes it look more significant, make the data look more significant, and so on and so forth. And unfortunately, while there might be other reasons why people have done it, it's just not acceptable, no matter how important it might have been for them, because many other research are based on the published data, and we are considering them as some sort of a standard, and therefore it is important that the ethics of it all is upheld, no matter how the results look. The last thing is the data privacy. When as bioinformaticians, one would be expected to maybe work with sensitive clinical information. So knowing what the regulations are is absolutely essential. Knowing that there are certain things that cannot be shared or put on the internet or used on some sort of a LLM to get information is absolutely essential. Please know your data, know your consortiums, know your rights and responsibilities before you get started. And last thing I missed out on explaining in the technical and soft management skill is the method of annotating your data. One can produce $1 million data and have poor annotation, and that the value of that particular data is almost nil. It loses all value if it's not properly annotated. The information about the data is much more valuable, because if that is not available, then unfortunately all the sequencing data is useless. So having a good practice of annotating your work, annotating your code, annotating your data set, all these things are vital. So please keep that in mind when you get started, as in your early years of bioinformatics, that these are all key points that can take you a long way when you actually start your own projects. So before I go into an example, do we have any questions? All right, then I will start with an example.
Yeah.
Yes.
Yes, I have one. So, like everything you told about refers to a person who wants to learn core bioinformatics. What about the people who have more wet lab but want to learn some maybe MD simulation things or GROMACS using or make a pipeline for predicting some mutation? How would people like us approach? Like, obviously, we can't do everything that you mentioned. So, how would we approach this problem?
Good question. Thank you for asking. I think I would just go back again and say collaboration is part of it. There are many wet lab biologists in my department, and we collaborate because there's always something that one learns from the other. And I would highly recommend collaborating with a bioinformatician. And that's usually what is recommended across forums. If you look at any bioinformatics forums, you can learn certain aspects. You can learn how a particular tool works. For example, Galaxy, that is a web-based tool for analysis, gives very clear instruction on how to use it for certain things. And if you follow it, you might probably get good results. And if you don't understand certain aspects, please use online forums like BioStars or certain Reddit pages where people explain if you have any questions about your data set or how to use it, how to use a particular tool, you can always look it up over there. And because there are the people who are managing it on BioStars are experts in their own fields and experts in bioinformatics, and they can definitely help out. But my recommendation would be to work in tandem with a bioinformatician if you have certain questions. And of course, there are many tools that require almost very minimal, almost no mathematical background, or as in like heavy mathematical background or coding skills, but that doesn't mean that it is okay to just jump in. Please ask for help. Please ask your colleagues if they can help out.
Thank you, ma'am. That was all right.
Yeah, so now I want to start with an example. This is from the consortium that we are that I am part of, and it's called the Solvardi Consortium, and that I've been working with actually. And we are or what we are focused on is on trying to understand and solve certain rare diseases that have been identified, that doesn't have a specific um, um, epigenetic or genetic cause of origin, which I will explain in some time. So the point being, the rare diseases make about 4.4% of the population, and there are many rare diseases that have been identified as something with genetic cause. So 70% of them have a genetic cause of origin, whereas about 30% of them have been associated either with epigenetic or unknown. We just don't know what's going on. And they have been identified as syndromes and not necessarily diseases as well. So these set of diseases or syndromes have been collected. There are a couple of them. I cannot give the exact number, but there are different networks in Europe that deal with specific set of diseases, and they've all come together, and in this consortium we try to identify the molecular cause of origin. These are the main goals of the Solvardi consortium, and to improve the diagnostics for rare disease patients. And the data set that we have been working on are genomic, transcriptomic, and epigenetic. So it's like across different omic layers. And we have we come from different parts of Europe, and then there's one North American collaborator as well in this particular consortium. And like I mentioned, there are those three data set, or the three omic layer that we've been focusing on. And we used, in my case, I just wanted to show that there was something called the RRBS. I'm assuming many of you know about this, whoever works on methylation data, that there's something called RRBS data set, which is the reason why I brought it over here is because this is one of the sequencing method that is available. We were using this as one of our ways of getting the methylation data. And the next thing I wanted to explain was, so I'm trying to find anchors from whatever we spoke before. Antara Sahar, I will answer the question right after. So the challenges I said was that it is very specific to each data set, right? So in my data set, or in this particular consortium data set, the problems are that it's a small sample size issue. Some of the rare diseases are just 1 in 10,000. So collection of these kind of data is very hard, and it can be complicated. And therefore, the sample size is essential, essential to be considered into my project analysis, my project overview, because depending upon the sample size, the power of my statistics can be affected. So one needs to be aware of that. And then the second one is the diverse nature of data, because the data comes from so many different places. You have the old, the young, with different set of syndromes and all these kind of things. So like understanding the data itself is a huge task. And that's why I keep saying that know your data, understand where your large variation comes from. Is it the sampling age, is the sample age, or are there any missing annotations in the file? Because these are all clinical samples. There might be, it's possible that there is not information because the patient forgot to fill certain things, or the patient's guardians forgot to fill something, and those things have to be accounted for, because they also have an effect on how you process your data after. and know your information as well. So now I mentioned that the data that I have are small sample sizes. So how do I identify with small sample size? It can be that just I have only one sample for that particular syndrome versus for all the 300 or 400 data that I have. So how am I supposed to identify? So it's not possible to use the usual methods. Like I can't just take a differential expression analysis and try to say condition A versus condition or control, because that doesn't work. So we had to come up or think about how to apply. So this is where you think you know your data and you come up with models, because or think about what is the underlying assumption. And if the underlying assumption over here in differential analysis is that there should be certain number of samples per group, then it's not fulfilling that assumption. So we have to think about another method. And here, the in this particular paper, Felix Brechmann, he talks about how a population can be taken, and from that, a single outlier can be identified. So that outlier is then looked further into and try to understand, and like there it's understood as to what's happening in that. Why is it that it's an outlier? Why are we having an outlier from the rest of the population, and then try to understand it. And that's what was done. And in this particular consortium, how we have done so far is to have a preparation phase, and this would be helpful for people who are in academic settings, who are in a place to have collaboration. So I recommend this method because we have done this so far. Which is to have a phase where people, the data analysis, bioinformaticians, the task force was created, and we had information that we analyzed together, and we put together information. We had the whole sequencing done and the analysis done, and then we all the different set of experts, from clinicians to biologists to technicians to geneticists and bioinformaticians, all of us got together in one place and had a discussion for a couple of days. Like we met and we tried to understand our data, tried to understand the results, because with communication, we learn from each other and we are able to, it's easier to solve or try to bridge the gap that is usually present between these different groups. And afterwards, we were able to follow up and see what was going on. And with this method, we were able to, the consortium was able to solve or understand what was happening for 28 of these rare disease cases that were collected. And so I want to emphasize on the advantages and the effects of doing collaborative analysis from different fields together, and the importance of it all, and how it is it can give certainly good results, even in terms of rare diseases. So do we have any questions before I go into my conclusionary remark? Or maybe I can finish it off and then we can go into questions. All right. So to conclude, what I want to do is a brief recap of whatever I've been discussing so far. First, we looked at the massive information load that is available at the moment, the speed at which bioinformatics has been evolving, it has been growing. And we were looking into how much of information is that is available at the moment. We also discussed about how bioinformatics is a complicated interdisciplinary field, which requires domain understanding in biology, informatics, in terms of mathematics, and also ethics. All these things play a huge role. And then lastly, I brought in an example to explain the data-specific challenges. Each data comes with its own set of challenges. Rare disease is different, cancer research is different, and epidemiological studies are completely different. So understanding your data and then trying to implement your workflow is essential. So if there's anything I would ask is that you take away these 3 points. The first one is that if you want to be a good bioinformatician and you want to get started early, I would say my biggest recommendation is to create consistent habits of learning. And doesn't matter if it's just bioinformatics; it's across all fields. And to have good data management, good ethics skills, and all these ethics and good data management skills. And the for the people who might be able to come together to have projects that are interdisciplinary, I would highly recommend that you consider interdisciplinary multi-omic approach for your projects, because that really gives the confidence that a particular disease or whatever we're studying gives that particular significance or not. And then the last one would be to implementing the 5 pillars of bioinformatics, to know that these are the core things that one needs to keep in mind. And in order to become good bioinformaticians, it comes with responsible and consistent work in when it comes to either coding or when it comes to understanding biology or whatever be it. It is consistency and ethical way of doing things. So with that, I would like to close my talk. Thank you so much for listening, and I will take the questions that are already there on the chat. And if you have any other questions, please jump in.
Thank you so much, Mrs. Midhuna, for your insightful talk. Now, we'd like to open the floor for the audiences to ask any questions that they have. You can use the chat box or turn on your mic directly and speak up. Thank you.
Hello, ma'am. I actually want to ask a question.
Mm-hmm. Please.
Yes, I have just written in chat a reference paper. If you can.
Hello. Okay, maybe I can just get started with one of the messages over here. Someone has asked if it is possible to go for a PhD in bioinformatics without having a hardcore knowledge in coding. I'm afraid no. It is necessary. If you're dealing with bioinformatics, it is necessary that you are aware of different tools, different statistical models. That is absolutely essential, I would say, when it comes to bioinformatics. As beginners interested in learning are any online courses. Online courses, I would say there are plenty. If your university or if you can afford to have a data camp course, there are multiple levels. You have the introductory lessons in art all the way to advanced courses. You can also find them on Coursera, edX, and also to take a book. There are multiple online markdown books. If you type introduction into R, there are multiple freely available content. And I would say another best way, once you're familiar with the basics, the best way to learn any programming language is to get started with a project. There are you can start coding with a particular project. If you're interested only uh, in bioinformatics related projects, I suggest Rosalind. Rosalind has multiple different data, and it would ask you to do something very specific. Like, for example, it would give a genomic sequence and ask to or whatever sequence, and then it was to reverse complement it or whatever be it, or divide it into, change it into FASTA format and things like that. So it gives you a way to learn on the with the project. So I would recommend something like that as well. Someone raised their hand.
Yes, ma'am, I raised.
Hi.
First of all, thank you, ma'am, for this wonderful lecture. It was really amazing. Yeah,
Thank you.
my question is that with the increasing complexity in and scale of epigenomic data sets, what role do you think that quantum computing will play in upcoming future research regarding bioinformatics and epigenomics?
Thank you for the question, Rahul. I must say I don't know. It's too early for me, and honestly, it's not within my sphere. That's what I would say. Um, I, um, it doesn't come into what I am working on at the moment. So, as well as I've not come across many papers or any blogs that talk much about the quantum computing in terms of bioinformatics. So I'm afraid I don't have much to say on that. So yeah, but thank you.
Thank you, ma'am.
Purvit?
Yes, sorry, I'm actually there was a network connection earlier. I was asking the
No worries,
question.
no worries. Mhm.
Regarding the reference that I've mentioned, like that GROMAX tool for drug library, if you can see in the chat box. Yes, I've sent it again.
Yeah, GROMAX tool, yes.
Yes. So, what specific prerequisites are required for me as a beginner bioinformatician to make something like this if I want to focus on AMR, antimicrobial resistance? So,
Thanks for the question, Purvit. I am not familiar with GROMAX. I'm assuming it's a complete package. Is it it's a Python or an R package, or what is it exactly?
it's basically used for MD simulations and prediction. Like this paper, Dr. Andrew Lin from JNU used this to predict mutations and further make drug conjugates.
Does it come with a UX, or is it something to do with coding, like you have to write a code?
I would say this comes with
UI.
a UX. Yeah.
Okay, it comes with a UI, like a user interface, and
Yes.
okay.
Basically, on servers, they have this to perform MD simulations and other things.
Okay, and your question is how to create a tool, something like something of that sort.
Yes. Like from a beginner perspective, what should
Okay.
my trajectory be?
Do you know what the underlying assumptions are, as in the sense, what is the method, or what is the mathematical background of this particular paper?
No.
Oh. So that's where it starts, right? So the
Oh.
UI interface is the outermost level. So you can create a nice UI, but what is it for? What is the basis on which this particular tool works? That, if one understands and then comes up with ways to write, that means you would have to write a code. You have to start writing code. You have to create a software package, and then come up with ways to host it on a server, and then that is transmitted across, like people can use it. So this is what bioinformatics, one of the parts of aspects of bioinformatics is. So one needs to know the basis on which this particular tool was built. What is it? What language was used? How did they? What is the mathematical model they've used? And how are they hosting it? What is the UI design? How did they put it together? So if you have understanding of all these things and you have your own idea, then it's possible. But that pretty much comes back to my talk, that you would have to have strong background in computer science and informatics and mathematics.
Sure. Thank you, Anand.
Yeah.
Very much. It was great.
All the best. Then we have some questions over here. Mohammed Jahid Khan, Rosalind. Yes, I think that's how it's pronounced, spelled at least, Rosalind. And Rohit Parekh, can epigenomics-based tools or platforms be developed as commercial products? There are already many epigenomics based tools and platforms that have been developed for commercial use. But usually when it comes to research, generally speaking, overwhelming number of packages that have been developed, it is developed as open source. And that's how it's been promoted so far. And of course, you can do it. If you have an idea and it's an original idea that you would like to patent and use for commercial purposes, of course. Yeah. And so we have two people who have raised their hands. Purvit is still, I'm assuming, not. Garima Mittal.
Good evening, ma'am. Thank you for the lecture. I have this question about if like I want to build a small project or like just get started in bioinformatics, then like and I don't have any prior experience with coding or other things. So like how do I get started in basic bioinformatic tools and databases? First question. Secondly, how do I find data sets? Like, how do I find data sets to like build a graph or something?
Okay, good question. First thing I would suggest is learning a particular um, programming language, either it's R or Python or any other language that is of your interest, Bash, whatever be it, learning it. There are many bioinformatics related projects online that you can find. If it is Python, you can get even more. There are many problems that are hosted online. You can have a look and then you can try to understand it. When it comes to data set that you would like to use it, I would suggest checking on SRA. So there are many NCBI related GenBank and others there where the data sets are being hosted. They are open, public access. So you can download them, try to understand. This is how I learned as well when I was doing my bachelor's, which is to explore, take time, read up on different there are multiple blogs that are available. Try to understand it, use and try to download a particular data set, try to understand how a FASTQ, a FASTA file looks like, and try to differentiate them, and try running the, for example, BLAST or on a NCBI, and try to understand how these different things work. And once you get a hang of it, you can create your own projects. Think about how to explore. These are all different small projects, right? So you're in the process of understanding different things. So please, I would recommend doing something like that.
Thank you, ma'am.
There was another question, or okay. Twinkle, what do you think an undergraduate should look out for in terms of internships? Should we start by giving working on our own guide or learning on our own? I would do all. So start, of course, learning on your own is important. You also have your own coursework that is available, that must be done without any issues. And then when it comes to internships, there are usually plenty of internships. It's usually hard, but you can start with where you are in the universities. There must be someone, if there's a bioinformatics department, ask one of the professors if you could work with them, gain some knowledge, and then you keep building your portfolio like that. Start small. If you're able to get one of those big internships in ICM or ICC, IISC, or NIMHANS, or any of the big places, if they have some sort of a bioinformatics program, internship program or something of that sort, please go ahead, try it all. But I would also say you have professors in your own places and in your own universities. Try to collaborate, try to ask for internships, and understand how they have done certain things. Of course, learning on the job is the best way to do it. Yeah.
Thank you, Mrs. Midhuna. I believe you must have received some questions from the registrations form. If you
Yes,
haven't,
that's right.
yeah, if you haven't addressed them, could you please address them now?
Sure.
Thank you.
I think many of the questions that were asked have been addressed in some sense, but if you feel like you haven't really understood it, please feel free to ask again. I will go through that. One question that I probably did not do is what role do machine learning and AI play in improving the interpretation of complex genomic patterns? A huge role, I must say. Machine learning, we have a lot of models. We're trying to understand it in our play. Several of my colleagues are trying to understand it from interpretable models. They're trying to develop interpretable models to understand the epigenomics. They are trying to. It's always good to have a strong foundation, and there is a need for it as well in some sense. Several papers are being published almost every day on different machine learning methods that are used for epigenomes. And I would say, yes, there's a huge need for it. The question is very general, so I'm going to leave it at that. I think it's good. There's a question about how can we use epigenomics for studying cancer cells and understanding signals that indicates cancer at initial stages. I'm sure there are many papers regarding that. Cancer biology is not my field, so unfortunately I cannot say from the top of my head, but I'm pretty sure there are multiple papers. Please try to read different papers. If you're interested in a particular field, look out for it. Type in epigenome cancer cells. You would be able to get multiple different publications on that.
Ma'am, I believe we have one more question in the chat.
Oh, thank you. As an undergraduate student beginning in bioinformatics, what is the realistic time frame for publishing a review paper on a defined bioinformatics workflow for a specific problem statement or disease? Please take your time. There's no rush. There are multiple reviews, but what you want to create is quality stuff. So in order to create quality information, read a lot, explore a lot, understand the key concepts, and then start writing. Because if it is only the beginning of bioinformatics, I'm not even sure what you mean by beginning into bioinformatics is. I would say please take your time to learn and then publish, because there's no rush, and there are plenty of, like I mentioned, plenty of available resources. So whatever information you want to add to the existing literature should be worthwhile your time, your energy, and should also benefit the community. So please take your time. Okay. There's a question on how does the integration of multi-omic data, genomics, transcriptomics, proteomics, epigenomics, and bioinformatics pipeline present unique computational and statistical challenges compared to analyzing a single data type, particularly concerning data normalization, dimensionality reduction, identifying causal relationships rather than mere correlations? Wow, that's a good question. Integration of multi-omic data, of course, comes with its own set of problems. Firstly, where does the data come from? Is it all from the same sample? Is it from the same? Is it from the same donor? If it's from the same donor, is it all from the same sample? If it is not, then do you see batch effects? If there is batch effects, are there any other technical effects that one needs to account for? All these things have to be considered, and not to mention storage issues, because you need high performing computers. One cannot just run it on laptops if it is huge data. So it is important to know. And when it comes to Um, you've mentioned about dimensionality reduction and others. Dimensionality reduction should be one of the basics one does in order to get an understanding of your data. So it's sort of like your QC, your exploratory analysis, to understand how the data looks like. So absolutely, it will change from data to data, and having a good understanding of how a normal or a clean data should look like is absolutely essential. If your samples are clustering by technical variabilities instead of the biological information, say for example, if you have instead of 5 cell types, it's clustering by batches, then you know there's batch effect that you need to account in your differentials and things like that. So these things are important, but it's a bit hard for me to give a straight answer because I don't know which data you're referring to or what you're working on at the moment. And but I hope this sort of helps. Thank you, Rahul. Appreciate it. You can always contact me over LinkedIn. If there's anything I could help out, I would try to yeah, share information there. All right. Please talk about CpG methylation also. I am not entirely sure what exactly you would want from specific CpG methylation. If it's just general overview or otherwise, because it's very, it's a huge topic. It can go for hours. If you have any, if you need specific information about it, or if you have any questions, please feel free to reach out on LinkedIn. In disease research, when does epigenomic analysis supported by bioinformatics support approaches provide insights that traditional genomic analysis might miss? Good question. Bioinformatics, your technical, what do you mean by epigenomic analysis versus the traditional genomic analysis, right? So the traditional genomic analysis, which is just taking your entire genome, you're looking at indels or some sort of a genomic mutation, and we're trying to understand what's going on. And you try to understand from that perspective. But then epigenomic is a layer above it. It controls how this particular genome is expressed or translated in, and and how proteins interact with the genome. So that particular level has a new information that complements the genomic data. So that's why having all these omic layers and studying them together makes more sense, gives a comprehensive understanding of what's going on than just taking the genomic level alone. Of course, it is informative, but it's taking the genomic level alone, but having it with the epigenomic layer is always more informative, and it gives the causality, the cause of a particular expression or whatever be it. Yeah.
Yes, ma'am. Any more questions you would like to address from the document?
All right. There's one question that apparently the general question for most registrants is, or maybe I'm mixing them up. There's a question about research and job opportunities in bioinformatics for students in Germany, and how bridge skill gap for industry and academia specifications as a beginner, indicating non-CS math and non-life science graduates, including, sorry. I would say having a strong portfolio, having really strong coding, mathematical background, and biological background. All the three spheres of bioinformatics, that is essential. One should have really good background on that, because the level at which masters would be taught is not basics. They're gonna assume that you have the uh, the domain knowledge, that is good, solid programming skills, solid mathematical skills, and all that. And once you have it, of course, you can start applying through to individual universities. You can look up for universities on this page called DAAD. It's a website of all the universities that website for, it's sort of like a database for all the universities in Germany, and it has all the public universities listed and their requirements for admissions. So please feel free to look into that and apply accordingly. One thing that I would really recommend for students who are considering to move to Germany is that you really think about not just the academic side of things, but also the culture shock that's associated. The first being, majority of the places in Germany are German speaking. Majority of the people are German speaking. So language is a huge deal. And the culture is different, language is different, and the climatic conditions, like the environment, all of it would be different. And if it is someone who's moving out from India or any other place for the first time, I would highly recommend that you are aware that it would be quite different, because it can have an impact on your education as well, because your one's ability to focus and to work can be affected. So please think through it, and if you are applying to a particular university, try to contact people from that place, or there are many online forums that are available. You can write to people, ask them if it makes sense to apply, what their experience has been in living in, and studying in that particular area and in the university respectively. So please do the research. It's not just the study part of things, but also the living part of things, because you will be expected to move from one place to another and start from scratch. So please consider them all. And if you have any specific questions, please write to me on LinkedIn and I'll try my best to answer.
Yes, ma'am. I have a question, if I may, please. I understand that the data that you're working with is exclusive to the experiments within your lab. And with regards to the consortium for rare diseases and such, you have the ability to modify the metadata in case it is incomplete, or get back to people who are working on the experiments for more annotation. But when we are working with publicly available data set, and there is a major gap, especially if you're considering a study that involves age-restrained sampling, and we need to analyze for a specific age and BMI or environmental factors, and there is a gap in the metadata that we're trying to analyze. How much do you think will it affect the actual outcome of our statistical analysis? And have you faced the same when you were working on your projects in your lab?
Yeah, thank you, Asiti. That's a good question. Yes, it will affect depending upon what information is missing. Say, for example, you are trying to understand, maybe you could give me a little bit more background. Is your data on transcriptomic level? What is it on? What kind of data
Yes,
are you
ma'am.
doing?
It was shotgun metagenomics for general transcripts for from all the samples. But the fact being that I was trying to screen patients who were less than 50 years of age for my study, and the majority of the data sets I was working with did not have that particular information. Despite the sequencing being complete and them having good quality scores, because I wasn't able to work with it, I think it might affect the quality of my analysis and the conclusions I derive from my workflow.
Okay.
because I would
Depending.
be missing out on a lot of recently available data.
I agree. Depending upon the type of missing data, say for example, the missing data is about is actually the age. Like you're missing certain age groups, and you instead of looking at all the age groups together, you want to stratify it and see it across in different intervals. If you missed age, then you pretty much lose that information. You cannot use it, and it's no longer useful. Or if there is a missing information on when it was sequenced, for example, that's even it's also a big problem, because if you don't have the information on when it was sequenced or how it was sequenced, if it is done on different batches, like in different batches or something, then that could also have a huge impact on what you are deriving as your result. What you are concluding as significant is probably just the batch effect for all that matters. So having that information is important, and I understand it, especially when it comes to secondary data. This is a complication for sure, but usually there is a corresponding author for all these data sets or paper that's written. And if you can contact them, write a simple email saying, hey, so and so, like, dear doctor, whoever, I'm working on this. I see that this information is missing. Is there a way to get this information? And then trying to explain what you've done so far, or what is lacking, or whatever it is, should be sufficient. I mean, like, usually in my experience, whenever there is a need for me to get a particular information, I've written to them, and depending upon how long they take, they usually write back, because people do want their work to be acknowledged or be used for something else. So my recommendation would be to identify what kind of information you're missing at the moment, and then the second thing is to write to the corresponding author.
Yes, ma'am. That's a very helpful insight. Thank you very much. I have one more question. Since you mentioned coding a lot and reproducibility with your analysis, um, right now when I am working on my project, I write a set of code to work on my data sets. And usually when we're uploading it to a repository, we will have to write down the README section of the files and give the appropriate data sets. So have you experienced limitations with regards to reproducibility of any of your coding projects that you have uploaded to your repository and such, where it was either limited by data or the version control of the algorithms that you have implemented within your code, and how can we overcome such errors when with regards to working on our own workflows?
Yeah, again, good question. When it comes to version control, or especially when you're dealing with multiple different projects or different libraries for the same project, right? I would highly recommend Fonda. Like, use some sort of a Docker or some sort of a Singularity, something. I usually use Conda environments to make sure that I have all the libraries within a specific. I make sure that the libraries are compatible to one another, as well as the YAML file that I write usually has. You can generate a YAML file from it, from the Conda environment, and that file could have all the versions of the tools that you're using, including the base tools, like for example just Python or whatever be it. All of it would be mentioned with the versions, so you could always add that as your as a supplementary file. And using the second thing would be to control randomness. So if you're using R, you need to set seed, so that there is no randomization that happens, because usually randomization is something that happens with every time you restart an R session. So use seed. There's something called set.seed, I think, and then you can have a particular number. So anyone who wants to replicate your work can set the same seed, use the same data with the same YAML file, and then try to run it. and most likely it would be it should be reproducible. And when we talk about reproducibility, in some sense, we're not as it's not necessary that it has to be like absolute 110%, like 100% of exact data match data results, because sometimes some of the tools have their own internal randomization process. So it is possible that one of the points might keep moving between the ones that especially that fall on the borderline of significant of 0.05, for example, can keep moving, shuttling between being significant and non-significant depending upon the randomization that happens. So that's why making sure that you have a seed set, having an environment like Conda environment exclusively for your data, the project that you're working on, and adding those information as a YAML file to your README, or like supplementary. You can't add it to your README, just a supplementary can go a long way. And using you can keep track of all these things on Git, so you should be able to do that. Yeah.
Yes, ma'am. And one last question that I have is, have you seen any major differences with regards to outcomes of your code when you're coding on your local node and cloud service? Does that affect the final compilation and posting of your repository and such? Not really.
Again, it depends on. No, it really comes down to which versioning. Like if it's the same versions, same set of information. Because I usually don't have the need to run my code on local machine.
Yes.
But I do know people who keep moving between these two, and they usually have the same versions. Like they make sure that they use the same YAML file to create a Conda environment, and then with that Conda environment they have installed, they have all their respective libraries, and made sure that it is consistent across. But at least like from the top of my mind, I cannot really say that it would make a big difference. But at least like that's where I would stand at the moment. Yeah.
Yes, ma'am. That was very helpful. Thank you very much, ma'am. And
Pleasure.
once again, thank you for taking the time to be here. This is the first time we're hosting a doctoral candidate, so I believe all the participants can relate more to your insights based on the proximity of our
I hope so.
experiences. Once again, ma'am, thank you for being here, and thank you for this wonderful lecture. And I'm sure a lot of our students from MSc have enjoyed it, because we're in the project semester. So we could relate your experience of having to get a new laptop for our projects and discovering new tools and softwares. Thank you very much, ma'am.
Well, my pleasure. And if you have questions, please write to me on LinkedIn. I might take some time, but when possible, I will get back to you with an answer. So thank you, everyone. Thank you for staying for, I don't know how long it's been. And